polymerase, the essential enzyme of transcription, is responsible for copying a DNA sequence into an RNA sequence during the process known as transcription. To initiate transcription, RNA polymerase and the necessary transcription factor must bind to the promoter sequence. The question that will be answered today is, what exactly are these necessary transcription factors? Enter the subunit of bacterial RNA polymerase, known as sigma factors.
So, what exactly is a sigma factor? As mentioned, sigma factors are the subunits that allow RNA polymerase to properly bind to the promoter site and initiate transcription. Since prokaryotes use the same RNA polymerase to transcribe all of their genes, sigma factors are able to regulate gene transcription by altering the RNA polymerase affinity for specific promoter sequences. This means that sigma factors determine the specificity of promoter DNA binding and control how efficiently the transcription is initiated. The sigma factor has three main roles in RNA synthesis initiation. 1. Target RNA polymerase to specific promoters. 2. Open regions of the double-stranded DNA promoter. 3. Interact with other transcription factors.
The sigma cycle and transcription's initiation. As mentioned earlier, the sigma subunit of prokaryotic RNA polymerase is what recognizes the consensus sequences found within the promoter region, the -10 and -35 regions upstream from the transcription start site, which allows the DNA polymerase to bind with specific promoters and allow for the expression of specific genes. It is the -35 consensus sequence, the TTG-ACG, that is directly recognized and bound by the sigma factor. Once this interaction is made, the subunits of the core enzyme are able to bind to the site. This binding allows for the 18-10 region to then facilitate the unwinding of the DNA template. As the initiation phase ends, elongation begins with the release of the sigma subunit from the RNA polymerase, which allows the core enzyme to proceed along the DNA template. This means that the binding between RNA polymerase and a sigma factor is transient. Or in other words, lasts only for a short time and is an impermanent bond between the two.
Hence, this sequence of steps is known as the sigma cycle. Without sigma, the core enzyme would transcribe from random sites on the DNA, as it is not being directed and bound to the promoter region, which would produce mRNA molecules that are unusable for protein synthesis. This is why, without sigma factors, there would be basal levels of transcription within the cell. Are all sigma factors the same? There are multiple sigma factors, each capable of transcribing a specific set of genes. All bacteria express at least one housekeeping primary protein, such as sigma 70 in E. coli. You'll want to remember this sigma factor for later. To explore this, let's take a closer look at the well-studied sigma factors of the bacteria Escherichia coli, more commonly known as E. coli.
E. coli, the most well-studied. E. coli has 7 sigma factors, each of which are responsible for transcribing a specific set of genes, the primary of which is sigma 70, a factor responsible for the expression of most, if not all, unconditionally essential genes. In other words, sigma 70 is the housekeeping factor. The other 6 factors are as follows: sigma 54, which is responsible for nitrogen metabolism, sigma S, which is responsible for the stationary phase, sigma 32, which is responsible for heat shock, sigma F, also known as sigma 28, which is responsible for flagellar proteins, sigma E, which is responsible for extreme heat shock, and sigma FeCl, which is responsible for ion transport within the bacteria. Having been the first of all sigma factors discovered, sigma 70 happens to remain the most well-studied factor, as well as holds the title of being the factor with the strongest interactions between itself and RNA polymerase. In fact, this interaction may actually be required for the sigma factor to bind to DNA.
Coli. What's so special about it? Sigma 70 is divided into a family of 4 sigma 70 factor groups based on protein domain structure and amino acid sequence, and act as the housekeeping sigma factor for E. coli. This housekeeping factor ensures that essential genes and pathways remain operational in the bacteria at all times. It is worth noting that while all bacterial cells will have a housekeeping sigma factor, the sigma factor itself will vary from bacteria to bacteria. In other words, sigma 70 is not always the housekeeping factor. Sigma 70, in the case of E. coli, is the primary sigma factor during exponential growth. It targets the RNA polymerase to a variety of promoters that are essential for normal growth. Changes such as heat shock or growth into stationary phase, which differ from typical bacterial growth conditions, lead to the replacement of sigma 70 with other sigma factors in the RNA polymerase holoenzyme complex. If this replacement occurs, the holoenzyme will no longer recognize the same transcription targets, which could result in a decline of RNA and protein levels.
With sigma factors comes anti-sigma factors. Anti-sigma factors are antagonists to sigma factors. When it comes to the regulation of gene expression in prokaryotic cells, the anti-sigma factors would bind to the sigma factors and inhibit transcriptional activity, as the sigma factors would no longer be able to bind to the core enzymes of RNA polymerase. The RNA polymerase cannot be directed to promoter boxes to allow for the expression of the required gene. If no transcription takes place, there is no mRNA sequence to be translated into the necessary proteins for the cell to function.